So again, hi there everyone, and today I want to talk to you about the RNA-Seq Rescue Algorithm. And again, my name is Saniya, and I really believe that this is a very important contribution to how we understand and work with RNA sequencing data. It's so important, we usually have a expression data for different genes, and RNA sequencing data is so cutting edge. And that's how we can come up with all these amazing discoveries that will touch all of our lives. So I just want to kind of dive into that a little bit today with an example of how you can work with the RNA-Seq Rescue Algorithm. Hi everyone, I'm Saniya. I'm the teaching assistant for the bioinformatics class, and today I want to talk about the RNA Rescue Algorithm. So typically, you're given RNA sequencing reads, possibly a genome sequence, and what you're asked to do is to, reconstruct full-length transcript sequences from that read, those reads of information that you get for RNA sequencing data. So we're focused on quantification. So, given the RNA sequencing reads and transcript sequences, can we try to estimate the relative abundances of, those transcripts, the gene expression, construct the gene expression data set. And that's important because gene expression data set and information is used for so much. I use it a lot of the time in my research work as well, particularly like for any data sets, like, neurodegenerative disease data sets, like, Alzheimer's, ADHD, ALS, Parkinson's, like schizophrenia, or even for cancers, lung brain data sets, all sorts of data. And RNA sequencing is very cutting edge. And we want to be able to, get that data, the gene expression data from this RNA sequencing data. And we can do differential expression, other downstream analysis, and, differential expression analysis will allow us to say that between two groups or multiple groups of individuals, some who may have the disease and others who may not have that disease, or maybe it's gradations of individuals, like very severe amount of the disease or not as severe, can we find the genes that are differentially expressed, that are differently, statistically significantly different between the two groups, for instance. So there's like so much that we can do with that information. And we want to use RNA sequencing reads from two different samples and transcript sequences to predict which transcripts have different abundances between the two samples. So RSEM is actually, it's a really cool algorithm that was developed. So right now I'm a Badger. I am at University of Wisconsin-Madison. And one of the professors here, Professor Colin Dewey, came up with the RSEM algorithm as well. He's one of the main authors on that paper. And that's helping us kind of figure out and deal with the fact that sometimes RNA sequencing reads do not always map uniquely to one specific gene or isoform. But it's super important. RSEM is basically RNA sequencing by expectation maximization. You don't need a reference genome, and it's able to help quantify those reads. So you usually have transcripts and read groups. And it's basically so important because RNA sequencing data is, transforming how we measure transcript abundances. And we don't know how to handle reads that map to multiple genes or isoforms. So what RSEM is doing is it's trying to help us deal with that. And expectation maximization can be very complicated to execute manually, but I want to kind of explain a little bit about it and work through an example that's called the RNA-Seq Rescue method. And that's going to help us use just one iteration of expectation maximization. And we're going to be working with a bipartite graph that has two types of nodes, transcripts and read groups. And we're just going to dive into that. So, just stay with me right now, and I'm going to walk you through a computational example of the RNA-Seq Rescue method. Thank you. And along the way, please let me know if you have any questions at all. So this is sort of like a little joke. So the DNA, which is always, double-stranded, is asking the RNA, hey, are you still single? And the RNA is like, always. So I just thought it would be a little bit of, fun humor. So little guy doesn't get to celebrate Valentine's Day much, I guess. And this is just another cartoon, and this is the central dogma of molecular biology, going from DNA to RNA to protein. And here I just sort of wanted to tell you a little bit about the different packages that are out there. Some that I have looked at as well for normalizing gene expression data are like, there's EB-Seq, and then there's DE-Seq as well. So EB-Seq was developed by one of the professors here, Professor Christina Kendziorski was one of them who was involved in EB-Seq. Then there's like DE-Seq as well. And sort of the idea, the idea is that when you're looking in, you see here, you have like treatment and then you have control groups as well. Then what you're sort of looking at is like, these are ensemble IDs, and you can map them to, gene IDs that we typically use, and then their associated entree IDs. And then there's the whole mapping process as well. The idea is that you will end up using and doing comparisons between them. Between the control group and those who have a treatment for a certain condition, for instance. And then you can find the genes that are differently or differentially expressed. And here, this is another example, using another RNA-Seq gene, another bioconductor package, more power to, the R. I really am very grateful to bioconductor. I've used it a ton. So bioconductor is really great. But you'll see here what they talk about is, for weakly expressed genes, we have, and I want to motivate you guys a little bit. For weakly expressed genes, we have no choice, no chance of seeing differential expression because the low read counts suffer from such high noises, and any biological effect is drowned in uncertainties from the sampling low rate. So what we can try to do, using some different normalization counts, so there's so many different issues out there with RNA-Seq data, and we're trying to detect the noise and being able to really uncover biological signals to try to make a difference. So that's why the methods that we're learning right now will be very helpful for better elucidating and understanding how do we deal with these issues when we get RNA sequencing data, and how do we really figure out the relative abundance of certain transcripts in our experiments. So again, I'm a happy Badger. I'm a graduate student at the University of Wisconsin-Madison. So I'm one of those Badgers. Go Badgers! So one of our professors at the school, Professor Colin Dewey, he's one of the authors on this paper and on this algorithm called RSEM. So RNA sequencing expectation-maximization, it's an accurate transcript quantification from RNA-Seq data with or without a reference genome. And I'll read this out loud. Because what we're going to talk about today will be the RNA-Seq rescue method to approximate one iteration of this expectation maximization step of RSEM. So what's important is that RNA-Seq is revolutionizing the way transcript abundances are being measured. But a key challenge in transcript quantification from RNA-Seq data is how do we handle reads that map to multiple genes or isoforms? So this issue is really important for how do we quantify with de novo transcriptome assemblies in the absence of sequenced genomes. It's really hard to determine what genes are isoforms of the same. So what transcripts are isoforms of the same gene? So do they belong to the same gene or not? And a second issue is how do we design RNA-Seq experiments in terms of the number of reads that we're using, the read length, and whether reads come from one or both ends of the cDNA fragments. How do we know how to do that? That's an issue that we're having. So a lot of times, a researcher like me, I'm going to be working with this RNA sequencing data, I'm going to be plugging it in for my analysis. Some of the packages or softwares that I've used are like BEC2, to do downstream analysis. But how do we get the data into the way that we want it to be? How do we do all those analysis? It's super important to understand these underlying algorithms that we use to take the data that we get and to make sense of that. And RSEM is a very valuable contribution. And it's very technically, a little bit advanced, and it's a little bit sophisticated. So we're just going to walk through an example for how we would do that. So RSEM, it's a user-friendly software package. You can quantify gene and isoform abundances from single-end or paired-end RNA-seq data. It gives all these intervals, and it enables accurate transcript quantification for species without sequenced genomes. It's got a pretty superior, performance. We can find the most accurate gene level abundance estimates with large numbers of short single-end reads. But if we want to estimate relative frequencies of isoforms within single genes, we can just use paired-end reads depending on the number of possible splice forms for each gene. And splice form kind of represents the fact that, when we have an mRNA, it's coming out of the cell nucleus, we could try to estimate like what that looks like if we have, let me try to sketch this a bit as well, but the key idea is that we would have, a cell and a nucleus, and coming out of that would be, so this is our cell here and our nucleus. So when we come out of here, our messenger, sorry, it's not quite coming out there, but it's like, you can imagine like inside the nucleus. I may be able to show it this way, that we have our whole cell over here, our messenger RNA here. This is our mRNA, and we have like, maybe we will have like an intron and exons, and it's all the splicing. So let me just say that, like, let's just pretend that this here, okay, so let's just say that these are the ones that we want. These are the exons 1, 2, 3. So what we could have for our processed RNAs, we could probably take 1 and 2 together. We could take 1 and 2 together, and that could be our final product, 1 and 2. So we would have like 1 and 2, like these are our exons, 1 and 2. Or we could have like 1, 2, 3. So like just to write it out, we could have these two come together. Maybe 3 does not get included, or maybe and this is like there's another like splice site in between them as well. You can pretend there's another splice site. So we could take 1 and 2 together. So I'll just list it out like this. So we could have like 1, 2. We could have exons together. We could have 1, 3. Maybe we would have 1, 2, 3. We could have like we could also have like 2, 3. Maybe we would just have them individually as well. So there are multiple different splice sites that are possible. 1 and 2 can come together, or maybe it's 1 and 3 can come together here, or maybe it's 1, 2, 3. We take all of them together, and this is another intron that gets spliced out. So the number of different splice forms that are possible. So RSEM is it's a user-friendly software tool. We can quantify transcript abundances from RNA-seq data. We don't need to have a reference genome. And it's enabled, cost-efficient design of quantification experiments with RNA-seq. It's currently relatively expensive. Now, what I look at is some of the data I'm looking at is post-mortem brain data. And one of the things we wish is that we could quantify data that, of gene expression values inside living individuals as they are developing neurodegenerative diseases, like such as Alzheimer's disease, schizophrenia, Parkinson's, and we don't have that information just yet. So what we're trying to do is so important with the existing technologies that we have to make the difference that we can and to constantly improve the metrics and the analytics that we can get from that data. That's what's really important. So here is this RSEM tool. So it's as accurate transcript quantification from RNA sequencing data with or without a reference genome. So you can understand here that this is like, you know, you have genome sequences or FASTA, GTF files, or you can have transcript sequences in a FASTA file. Then you can use RSEM for prepare and reference, RSEM references. You could also have the RNA reads FASTA, FASTQ files as well. You can calculate the expression. So this is all that's going on. This is like expectation maximization at play. And Professor Dewey has on his GitHub site over here, deweylab.github.io/rsm, he has RNA-Seq by expectation maximization. And there's a lot of code here that you can use as well. And there's a lot that you can do to develop and to contribute to these efforts to improve the insights that we can get from such data. So credit to Professor Daifan Wang for this slide from his Advanced Bioinformatics course. So how do we currently, how do we quantify with our RNA sequencing data? So we can suppose that reads are of length 1. Typically, there are about 35 bases. So just pretend that, this is just, 1. But typically you would expect maybe a read could be like, A, T, G, C, C, or something like, 35 different bases in this read. But right now, to just keep it like, it could be anything. Like we could also have maybe another T as well. T, A, T, G, C, C. We could just add, like, typically there are about 35 bases. But for now, we're just going to pretend that there's just one read here. One read, these are the reads of the lengths. So what relative abundances would you estimate for these genes over here? Relative abundances is the relative transcript levels in the cell, not proportion of observed reads. That's something to keep in mind. So it's the relative transcript levels in the cell. Out of all the transcripts that we have, it's not the proportion of observed reads, it's the relative transcript levels inside that cell. And again, typically these are greater than 35 bases, but for now we're just going to say A, G, C, G. These are, what they're mapped to. So there's a length dependence. The probability of a read coming from a transcript is proportional. So this line means proportional to the relative length and relative abundance times the length over here. So transcript 1 relative abundance would come from here. So let me highlight this maybe and show this part here. This 100 part here comes over here in the numerator, and this part here, this 200, will come over here. That's the transcript 1 length. And this yellow part is a probability of a read. This part here is a probability of read from transcript 1. So we look at all these reads here, 100, 60, and 40, and we're going to look at what is the probability that we get the read from transcript 1. So I'm going to say, okay, well, there are 100 reads here, and how many reads do I have in total? Well, I guess it's a bit confusing here, but like just say 100 plus 60 plus 40, which is equal to 200. So that's where we get this 200 from. So it's 100 out of 200 of these total reads belongs to, belongs to A over here. So 100 out of 200 belongs here. So the probability of reads from transcript 1 will be 100 over 200. That's what this is, 100 reads divided by 200 total reads, and then you divide it by the transcript length for 1. So it's 100 out of the 200 total, then divide this whole thing by 200, which is the transcript length for 1. And similarly, that's how you can, and that would be equal to 1 out of 400. So that's a relative abundance of read 1. And you can do the same thing here. So we look at the probability. So again, 200, 200, 200, we get from summing these up over here. So when we sum them up, we get 200. So that's where we get this value here. 200, 200, and 200 here. That's where we get these 200, 200, 200 values from. And that's where we get the 200 from. Then we also have to look at is this transcript length. So for 1, it's 200. That's where the 200 comes in the denominator. For 2, it's 60. So that's where this comes from. And for 3, it's 80. So that's where this comes from over here. And our numerator is going to then pick up on this. So this, like, let's just use sequence 3, for instance. This is a 40 here. This 40 comes from our G. This is the number of reads. So out of the total number of reads, how many of them belong to this? So this is a G, but you know, sorry, kind of wipe this part out. This is, yeah, this, let me just, maybe I highlight it this way, yeah, 40, yeah, here. So that's what that's 40 out of 200, you know, for the third group. And then you divide it over by 80, which is this length for transcript 3. So 40 out of 200 will be the numerator, and the denominator will just be this 80. Similarly for this, you know, transcript 2, relative abundance, 60 out of 200. So this is the 60 here. Oh no, sorry, this is the 60 over here, divided by 200. This will give you the numerator. So your numerator will come from your reads, in a way. Numerator is from the reads. The denominator comes from the transcripts. So this is 60 out of 200. Then you divide this by 60 over here, the 60 over here, and then you're going to get 1 over 200. And again, like for transcript 1, we get the 100 out of 200. So the numerator gives us the reads, and the denominator is going to be this 200 here. So we get these values, and these are like the relative abundances of transcript 1, 2, and 3, but we need them to sum up to 1, because, you know, they have to add to 1. So we're going to just normalize these so that we scale them up. And what you'll notice is that effectively this is just the same thing as 2 out of 400. So this is like twice. This is twice the abundance. So this is like twice this value, you know, it's twice of that. So that's why in the end, when we normalize them to get them to sum up to 1, we're going to get this to be 25%, this to be 25% for transcript 3, and this to be 50%. So it's a little likelier that we're going to observe, you know, transcript 2. So here we're going to talk about a specific example of how we use the RNA-Seq Rescue method. So this here is a bipartite graph. So we have these 2 types of nodes. We have the transcripts here that look like rectangles, and we have the read groups here that are the circles. So let me just kind of note that here. So we have our transcripts, which are the rectangles. Eeks, I can't sketch a rectangle. At least I'm not a square, so yeah, I don't know if we get the pun, but okay, anyways. Well, so we have transcripts, and then we have our read groups here, and try to draw a circle. I'm gonna make a smiley. Okay, someone's a smiley. Okay, so we have these 2 types of nodes. We have our transcripts here, and then we have, you know, our transcripts, and then we have our read groups. It is a bipartite graph, and this is the example we'll be working on today together. And again, along the way, please let me know if you have any questions at all. So the transcript nodes, they contain a transcript ID and the transcript length in base pairs. So you see here, transcript 1, 40 base pairs. Transcript 2, 270 base pairs, you know, the DNA, where, you know, it can be A, T, G, C. Transcript 3 is 1,000 base pairs. Transcript 4 is 120 base pairs. And transcript X is 100 base pairs over here. So that's what the transcript nodes refer to. And the read nodes contain the read counts for a group of reads that all align to the same transcripts. So there's 620 here, 390, 405, 4100, 5500, 40, 180. And these edges are designating the transcripts to which each read group aligns. So as I mentioned, the edges are going to designate the transcripts to which each read group aligns. So, as we talked about, like, we have the transcripts here, and then we have our read groups here. Yeah, getting better at this, huh? Okay, so like, let's say that we're going to focus on transcript 1, okay? So transcript 1, how we would interpret this edge, so it aligns to this read, it's the only one that aligns to this read group 620 over here. But it also aligns to this read group here that's, 390, so it aligns here. Okay, and you'll see in just a second, I filled about half of it. So transcript 2, it's the only one here that aligns to 405. So it's got it all to itself, 405 over here. But then it's also aligning here to this, 4100. Now, let's kind of, I know this is going to be a little bit hard to read. So transcript 3, it also happens to align to the same read group of 390. So this is shared. So it's shared between the two. It's also happening to align with this read group here, which is, by the 4100 that's shared with transcript 2. So this is almost like becoming purple, red, yeah, sorry, it's kind of like becoming all purpley and a little bit messy as well. This is purple here because they're shared here between red and blue makes purple, and this is becoming orange. It's shared between these two read groups. Transcript 1 and transcript 3 are sharing this 390 is shared by transcript 3 and by transcript 1. Transcript 3 also has its own little guy too. It has it all to itself, this 5500 all to itself here. 5500, it has it all to itself here. So transcript 3 again has 5500 all to itself, but it's also sharing this 4100. I know it's a little bit hard to read, I'm sorry, with transcript 2, and also it's sharing 390 with transcript 1. So that makes orange red, and yellow makes orange, red and blue makes purple. So you can kind of see this over here. And not only that, but then you also have to worry about, okay, well, transcript 4 is also joining this mix too. So it seems like a lot of them are sharing this 41, 800. Maybe I shouldn't have used this color. Maybe like how about I just like kind of walk you guys through it a little bit slower again. Anyway, so pretty much let's just take it again. Now it looks a little bit cluttered. So we have transcript 1. Transcript 1 is just got this all to itself. And this is when I will talk about kind of like the unique mappings that we're looking at. Transcript 2 has this all to itself here, this 405. Transcript 3 has this right here. This is transcript 3. And not as many colors, I'm sorry, to choose from. So I'm just like, I know like I've used red and blue a lot here. Also, red's my favorite color as of now. So so here we also have this purpley color here. Like, just ignore the mixing. This is transcript 4 here. It's aligned here to this 440. And then like maybe I'll use a light green here for transcript X. It's got this all to itself. In fact, it's not even sharing anything with anyone. It's all by itself. Then we can kind of track, these are like the mixings. Like, so this is belong uniquely to transcript 4, uniquely to transcript X, uniquely to transcript 3, uniquely to transcript 2, uniquely to transcript 1. And then these are the ones that they kind of are sharing between transcript 1 and transcript 3, this 390. Transcript 2, 3, and 4 happen to share this 4100. So this is showing you kind of how you can use a graphical representation to kind of explain this information about what is being shared between the transcripts and the read groups based on what they align to, based on like the sequence information as well. And we're going to try to figure out, relative abundances of these transcripts based on this information. So our task is to please use the RNA-Seq rescue method to calculate the relative abundance for these 5 transcripts here, 1, 2, 3, 4, and X. So these are the 5 transcripts that we have. And we have the transcripts and the read groups in this bipartite graph. So this is sort of showing a little bit about like RNA sequencing data. Got this from online. I looked up RNA-seq data. I just wanted to kind of show you an example of kind of like what RNA sequencing data usually is. So we have like the samples of interest, a condition that, like a health condition, like a tumor that we're looking at. It's a pretty sad state here. And then we can have a normal condition. So individuals or cell lines, or it can be as granular or as population level as you want it to be. But you have your data of interest. So at least two different groups. One of them will have the condition of interest, like a tumor, or and one normal usually. So we isolate the RNAs, ribonucleic acid. We have a poly-A tail at the end of them as well. So we generate like the cDNA fragment size, add linkers, we get the sequence ends, hundreds of millions of paired reads, tens of bases of billions of bases, of sequence. In the RNAs, it's adenine, guanine, G, uracil, U, and cytosine, C. So instead of thymine, you have your uracil. Then you have your unsequenced RNA, you have your RNA reads and short inserts. Then you can try to map to genome, transcriptome, and predicted exon junctions as well. And you could have many of these aligning to many different genes. Some sequences could be common between them. And even the same gene can have different variants. That's alternative splicing. That's why we're very complicated. And I think that we may even have a lot less protein coding regions than other less complicated organisms as well, because we have a very complicated system of alternative splicing. So we can have same region, like different introns and we get introns and exons, but it's a combination of exons that we splice together, and it can be different. And that gives us a lot of genetic diversity as well. So we want to figure out, predicted exon junctions as well, map to a genome, a transcriptome, figure this out, because this is our current technology for how we can sequence the expression of various genes. So then we can do our downstream analysis as well. So this is RNA sequencing data that we're having, and we're trying to predict what is the relative abundance of a certain gene in a certain condition. What bulk RNA sequencing is usually, it's looking at within individuals in a population usually. So you might have individuals who have like lung cancer, or individuals with diabetes, and then you're comparing them with individuals who don't have it. So you'll have like two populations. Or with single cell data, so it's more like an average across different cells. Like there might be different lung cells. Or if you're looking at a brain condition like Alzheimer's disease, well, we know that we have different brain cell types. Like we have neurons, but they're not the only type of cells in the brain. We can have oligodendrocytes, we can have microglia, which are the resident immune macrophage cells of the brain, and they're implicated in a lot of diseases right now, like neurodegenerative diseases like Alzheimer's, for instance. So we also have oligodendrocytes, we have astrocytes, all these other cells that are in our brain. So single cell RNA sequencing data is even more cutting edge, where we can kind of analyze and hone in on those specific cell types, because they're so different. Even in our immune system, we have macrophages, we have neutrophils, we've got T cells, B cells, we've got all sorts. For instance, we've got a lot of different types of cells, and they each do different things, and the immune system is incredibly complicated. So what we want to do is we want to try to be able to use this to do our downstream analysis. And this is another example that with the RNA sequencing data, you might have like different experiments here, or different individuals or samples, and you have your gene data here. And you could figure out the genes that are upregulated, that are increased in a disease condition, or that are downregulated, decreased in the gene condition. You can run enrichments, do all sorts of analysis based on this information. So RNA sequencing information, but as the people are saying, garbage in, garbage out. If we have garbage data to work with, sadly, I don't mean to be condescending, but this is a generic term, garbage in, garbage out. If we don't have good data to start with, our results may be erroneous or may not be as good. We might not even be able to detect the true signal that there is. So these methods like RSEM and understanding RSEM and RNA-Seq rescue methods are very important to be able to get the most out of our data that we are gathering. And hopefully these technologies will improve. But currently this is what we have, and we have to use these computational methods. And maybe you could be one of those people to help innovate in this field. So I kind of wanted to use this example to also just show you a little bit again about, the different types of RNA sequencing data applications. And this SC refers to single cell RNA-Seq, and bulk is bulk RNA-Seq. So it's from different individuals, samples. So these can be samples, many different individuals. They could have like the disease, or they may not have the disease. And there can be the control of various stages of a disease. And then these are genes. So this is an expression level matrix. So it's more like an average. So, for a brain tissue, for instance, if you look in the hippocampus, or if we look at the dentate gyrus, or if we look at the prefrontal cortex, it may be an average of the different cell types there, but we know that who it belongs to. That person might have schizophrenia, or maybe they have autism, or amyotrophic lateral sclerosis. Remember, you had the ice water bucket challenge, dumping the cold water, raising awareness for ALS, Lou Gehrig's disease, which Stephen Hawking had. These are very tragic conditions. So, but in basic, you just know if the person has the condition or not, if that sample had it or not. At the time right now, we can usually take it post-mortem for individuals who've passed away, but we can then figure out, like the genes by samples, and it usually is an averaging approach. So what we can figure out is we can, look inside those samples, but we may not get like as granular level data as with single cell. Single cell allows us, we know the cell types, and then the gene information, like we can tell if it's a neuronal cell, if it's a microglia cell, if it's what all cells that these are, and we can then do more a granular level. And oftentimes what these will look like is these will be dimension reduction and clustering, like this t-SNE, which is stochastic neighbor embedding. We use dimensionality reduction as well, figure out the cell types. For instance, we can do network inference here. For here, we might just look like, we might just try to figure that thing out. We can often, go from bulk RNA-Seq to trying to figure out single cell to single cell deconvolution. There are a lot of different approaches. Currently, a lot of my work has been mainly right now with bulk RNA-Seq, but I hope to be able to understand and better apply single cell RNA-Seq data as well. And I'll keep you posted on that. But for instance, why is it important again, single cell RNA sequencing data can be important because oftentimes if we just take average data from bulk RNA-Seq for epithelial cells, there's so many different types. There's squamous, cuboidal, columnar, connective tissue, and there might be like a lot more, I don't even know. But these cells, they form the lining of the surfaces of the body. They're found on skin, blood vessels, urinary tract, and organs. Then the fibroblast cells, they make up most of the connective tissue. They help sculpt most of an organism, provide structural framework like stroma for tissues, and are involved in wound healing. Then we have these immune cells, that they're involved in a network of biological processes to help us from disease. They respond to pathogens, viruses, worms, all that. I know right now it's coronavirus. It's, we all are worried about the immune system and our immunity, right? And it's also involved in our response to cancer. And recently, even we're realizing that like schizophrenia could even be like an autoimmune disease. So the B lymphocytes, the neutrophils, monocytes, T lymphocytes, mast cells, these are like antibodies, these Y regions, these immunoglobulins. This is a B cell, and its main job is to produce those antibodies. Dendritic cells, antigen-presenting cells, T lymphocytes, eosinophils, monocytes, basophils, macrophages. These are phagocytes that, they eat up as well. And these, antibodies can make those pathogens look even yummier to these macrophages through a process like opsonization, where they, attach to the pathogens, and then they make them look even yummier to the macrophages. But I just kind of want to let you know that there's so many different cell types, and if you're looking at an average of, these regions, you might not be able to pick up on specific differences that could happen. So that's why there's like also a big move towards single cell RNA sequencing data. But then again, there are also going to be those methods to be able to better figure out, what those are. Currently, even in the brain, it's a little bit difficult to measure microglia cell types, for instance. And a lot of this data, people are trying to figure out, can we go from bulk RNA-seq to figure out those cell types? Like, how can we also work with what we have and make that better? And that's where understanding methods like RSEM and how these work can be very important. And even here, I just want to give you another example, in a cartoon example. Like, let's say we're looking at ulcerative colitis. We could have healthy colon. We could have a non-inflamed colon, part of it for a patient. And then we can have an inflamed colon for the same patient. And we can have this healthy as well. And, healthy, non-inflamed, and then inflamed, different severities. And we might want to compare healthy with the non-inflamed. It seems like it's going to get inflamed, but it's not, like in a patient, maybe part of it is inflamed, the other part is not inflamed right now. So we might want to look at the part that's healthy in a healthy person without ulcerative colitis, with a person who has ulcerative colitis, but it's a part of the colon that's not inflamed. We might want to do that comparison here. Or maybe for the same patient with ulcerative colitis, the non-inflamed with the inflamed parts, non-inflamed with the inflamed parts. And then here, the healthy individual with non-ulcerative colitis with the inflamed part of the colon with the person with ulcerative colitis. So we can compare the two extremes, or we can compare the pairwise, and like looking at the increasing severity of ulcerative colitis. So the first thing that we want to do is we want to represent the graph, in terms of our read groups to our transcripts. So that's one thing, these are some of the approaches that I would use. So let me just try to break it down in this way. So, we go from our read groups to the transcripts. So the first thing we're going to say, okay, well, 620, let's kind of go backwards and say, okay, well, where does that come from? So the 620 over here, it comes from, and we kind of talked about this before, it comes right from transcript 1. So that's where we get this from here, the 620 to transcript 1. Then the next thing we're going to say is, okay, well, let's focus on our 390. This 390, it comes right from transcript 1. So it comes from transcript 1, but it also comes from transcript 3. So that's where we get this from, transcript 1 and transcript 3. They both have their edges that are coming to 390. So this 390 and transcript 3 to 390 here. Then we look here and we find that 405 part here, the next one in line is 405, and it comes right from our transcript 2. So this is what we find over here. Then we're going to find this big one here, this 4100 one here. So we find that this 4100, it comes from transcript 2, right? So this comes from transcript 2 over here. I'm not going over it on top of it because it'll, it will be difficult now to read it. And it's also coming from transcript 3, where we get this from. And it's also coming from our transcript 4, which is where we get this from. And in terms of this, I'll have it like this so it looks a little bit easier to understand this. Then what we will say is that this 5500, this 5500 one, is coming only from transcript 3. So that's where this comes from over here. And we can just say that, transcript 4, it's the one that's reaching to this 40. So actually we could have gone for 40 is coming from transcript 4. So sorry, 40 coming from transcript 4. And last but not least, this, for this value here of 180, this value right here of 180, it's coming from our transcript X. And that's where we get this from here. So that's how we can basically take this, graph apart and sort of look here and try to do this mapping. And what you're going to notice we're going to do next is we're going to say, okay, well, this has like 1 transcript group, this has 2, this has 3. So we can kind of, in a way, just go down this and say that this is, this will be 1 for this one. This will be 2 transcript groups. This will be 1. Oh, let me write it here. Like 1. This will be 3 groups here. This is 3. The thing is here. This is 3 groups. This is just 1 transcript group of 3. This is just 1, which is transcript 4, and this is just 1. So what you're going to notice is as we go, we can map these read groups here. So we just gone from our read groups to our transcripts. So again, 620 goes to our transcript 1. It's only coming from transcript 1. But this 390 is coming from both transcripts 1 and 3. So transcript 1 and then transcript 3. So that's why we had these here. 405 is only coming from transcript 1. So it's just having this 1 transcript. That's why I put a 1 over here. 4100 is coming from 3 transcripts, 2, 3, and 4. That's why I have it looking like this, and it has these 3 transcripts. Actually got a lot of these transcripts here. 5500 is only coming from transcript 3, so this is a 1, and all these 3 remaining ones are only coming from 1 of the transcript group. So that's why these counts are 1. And you're going to notice that what we're going to pay attention to, what's going to be what will make us happy to begin with, will be, so we're going to keep this graph, but what we're going to focus on is we're going to focus on this one, on the ones that have 1s that are unique in the next step. So that means that we're going to focus on these. We're going to focus on this one over here and this one. So I'm going to go in a bit neater like fashion on the next slide. Not that great at this, guys. Still not that good. So then what we're going to do here is we're just going to find the ones that only mapped to 1 of the transcripts. So again, what this would be is that this one is the one that had 1 of them, only transcript 1. This one only has transcript 2, 4, 5. 5500 transcript 3, 40 transcript 4, and 180 is transcript X. So those are the reads, or transcript lengths that are uniquely mapped. That means that they just have 1 of the transcripts that they belong to, just 1. So that's what these are. These are all just having 1, 1, 1, 1, 1, and 1, 1. That's where we get these from. And that's where you'll see that we will just focus on these. And that's why. So as you see here now, we found the reads and transcript lengths for uniquely mapped reads. So what we'd found is that 620, this read group over here, comes from transcript 1, which is what we found. We found that 405 read group comes from transcript 2, 5500 comes from transcript 3, 40 comes from transcript 4, and this last one of 180 comes from our transcript X. So we now can put it into this table, where we just focused on these unique mapped groups. So all this is over here is this, we just created a table based off of this. That's what this is, all of the unique read groups for each transcript, which is very important here for the RNA sequence rescue algorithm. So then we also need to next move. So what we did again is we had calculated all of these, unique groups again, as we just did this right now. And again, please ask me if you have any questions. So we just need to kind of just connect the lengths of each of these transcripts. So for transcript 1, we see that it's, so it's the same table as before, but we've just added an extra column of transcript length in base pairs. So transcript 1 has 40 base pairs here. So we just basically just take these values here. It's 40 base pairs, and then we just map each of them here, for instance. So this 270 for transcript 2 is here. So we just put the corresponding transcript lengths for each of these transcripts over here that we have. So 120 for transcript 4 comes here, and then this 100 for transcript X comes over here. So now we have the unique groups, and we also have the transcript lengths in base pairs. So then in step 4, what we do is we find the unique read group for a transcript, and then we divide it by the transcript length. So we just take those two columns, so this over here, which is just the unique read groups, and then we divide them. So, 620 again coming from transcript 1. So we just take the 620 and then we divide it by 40. So this is the 620 over here, and it's uniquely mapped to transcript 1. And we're just going to divide it by the 40 base pairs. That is the transcript length for transcript 1. So that's what we will do over here. 620 divided by the 620 here, which is this unique read group for transcript 1. So all the unique read groups that map for this transcript. And then we divide it by 40, which is the transcript length for transcript 1. And then we get 15.5. So that's how we do this. So if we had like another, like for instance, if we had gotten like another read group that happened to be unique for transcript 1, like let's say we had another one like this, like we can just, let's say that this is like 11. We could have just added like 620 plus 11, and then we would have had that's 631. Very low count, but then we would have gotten like 631. Like the numerator would have been 61. And then you divide 631 by 40. So that just kind of shows if it's just mapped to something new, that's sort of how you would kind of add that in. I thought that might be helpful. Because sometimes even for me, I'm always like, okay, well, what if, something is different? How would you, troubleshoot for that? I'm just showing you, you just have to find the unique ones for each transcript group. And then divide them. So that's why here, what you'll see is 405 is for transcript 2. And then that's why we divide that by the 270 again. So we just take 405 divided by 270 and we get like 1.5. So we just basically are taking this. And we're dividing this column, the unique read group, by the transcript length. And then we're getting this part over here. So we're just dividing 5,500 by 1,000, and we're getting 5.5 for transcript 3. In transcript 4, it's 40 over 120, which is 1/3 or approximately 0.33. And then 180 divided by 100 is 1.8. And then you can take these values here and then you can add them up. You just have to add these values up. All these values, you just add them up. And then you're going to get 24.63. So once you add all of these up together. So you'll see is once you have added them up, the sum is going to be 24.63. And that's what I just showed here, just the same thing, just simplified. And now you have the unique read group, transcript length, and then this is the set of values of unique read group for transcript divided by the transcript length. So we were just focusing in step 4 on like trying to find out the unique parts that correspond for each of the transcripts. So the next thing we want to do is we want to, in step 5, find the unique transcript relative abundance value. So F transcript for like a given transcript unique. So what you'll do is let's focus on, these are the same values we just computed, adding up to 24.63. So if we want to find the unique transcript relative abundance for transcript 1, what we will do is we just have to take, put this into a fraction out of the total. So you just have to take this value here and then just divide it by the total. So it's just like a, like a kind of like a percent out of 100. So it's a relative value. So we take this 15.5 and then we divide it by our total, which is 24.63, and then we'll get 0.62923. So that is the unique transcript relative abundance for transcript 1. So it kind of tells us, on its own, how much does each transcript, like what is the relative abundance when we figure out which read groups it maps to on its own, like what's the relative abundance that we find. So that's sort of how we will compute this. So that's how we found F 1 unique is 0.62923, and then we just add it to this column here, and we can do the same thing for the others. So here we will just take like 1.5 divided by 24.923, and that will give us this F2 unique. So that's sort of what I did here. So over here, you'll just see is that we just take, the next examples, we just take this 1.5 here, and then we just divide everything by this common total value here, which is 24. You can see there's 24, 24.63, 24.63. So it's just like the percent out of like 100. So what you'll see here is, as expected, these values should add up to 1. So this is like 62.923%, which is quite a lot, like a huge amount. And this is like around 6%, okay, not as great. This is 22%, 0.3%, so it's about the second highest, unique transcript relative abundance value. Then we have, like about 1.35, so pretty low for transcript 4. Slightly higher than transcript 2 for this transcript X, about 7.3. But yeah, again, it's just, you take this, unique read group for a transcript divided by the transcript length. You just take this value and you just take it out of the total. So, you just take these values out of the total. And that's sort of how you calculate your, your value for unique transcript relative abundance values for transcript X as well. This 1.8 divided by the total. So all these values would sum up to 1 if you add them up together, so that you can think of them like percentages in a way. And it's sort of telling you that transcript 1 has like a much higher unique transcript relative abundance overall. So that's just a simplified version of this table again. So you can see these unique transcript relative abundance values, and then they sum up to 1. So in step 6, we want to update the counts for each transcript. So when we think about it this way, when you are looking in a particular transcript, for instance, like let's focus on the first one, on a given transcript. How would we decompose this together? Well, what we're going to see is that a given transcript here is, transcript 1. What it has is it's going to have some parts that are unique to it, which is what we were looking at right now. We saw that it has the 620 group that is unique to it. This is unique for it. The 620 is unique for, group 1. But then it's also it could also have some parts that it shares, or maybe it doesn't, but it really depends. So it could have like a part that it's sharing, which is this, 390, which it happens to share with another group that here is transcript group 3. As you see here, transcript 3 shares this 390. So the shared counts with the transcript number 1 is that we find that it has 390 here for its shared counts, and its unique counts is 620. So sort of like, let me just take a little toy example, make it a little bit easier to understand as well. So for transcript A, we have like the counts here. Transcript B, we also have like, those counts, and, D. But what really matters is not the counts right now, because we sort of have them kind of covered into this, relative unique abundance counts we just calculated. These are the values we just finished calculating on, that we just found. So what we have is that this, if we are looking at transcript A, if we want to find, the C rescue for transcript A, then what we have to do, so let's just focus on transcript A. Okay, so this would be for transcript A. And this is also for transcript A. Okay, so like let's just focus on transcript A here. So for transcript A, what we're going to see is that, like if we are looking at its counts, if we're trying to update its counts, what we will find is that, the shared counts for transcript A. Okay, well, the thing we notice, okay, so first let's start off with the counts will be the unique counts for transcript A, like here. So for transcript 1, we saw its unique counts is 620. And then it has shared counts here, like, 390, which it happens to be sharing with transcript 3. So similarly, in this toy example, like a nice little toy example, we have transcript A, and transcript A has its unique count of UA. So that's what its unique count for transcript A is UA. Okay, and here, like for instance, it could be 620 for transcript 1. And now, like, let's say, just to make it a little more complicated, that it happens to share multiple groups. So you can kind of think of multiple groups as being shared with like transcript 3, for instance, a 390 with transcript 1, and this 4100 with transcripts 2 and 4. But in any case, I'm just showing you that it can be as complicated as you'd like. I mean, I don't know if you'd like it, but you know, in the real world as well, it could be very complicated. So you have transcript A that has a unique count of UA, but it's also sharing read groups with transcripts B and D. So SAB refers to the read group shared by transcript A and B. And SAD, SAD, huh? Okay, it refers to the read group being shared by transcripts A and D. So that's what we're going to notice, that if we want to find the shared counts as well, we're going to need not just the unique counts of UA, we also need to take into account the shared counts, but we need to scale it in a way. So we have that they so transcript A shares a read group SAB with transcript B. So the shared counts that we would take in, what does A get from it? So if they're sharing this value, like for instance, like transcript 1 and 3 are sharing 390, but here transcript A and B are sharing this SAB, what part will A get? Well, A will get its relative fraction. You know, if we look at those unique abundances, we can take A's fraction out of what A and B get. So it's sort of like, you know, you're scaling it based on the total that A is getting, the relative unique abundances for A divided by the unique abundances for A and B. So sort of like it's scaling it based on how the unique abundance of A is relative to B. This is the scaling, and then you just take those counts, and it's like how much does A get out of those counts? It's like a scaling, like a weighted average, and it's sort of like the weighted average idea in a way. It's like you're just scaling it based on those relative unique abundances that we just calculated. So transcript A will also do the same thing with transcript D. So you're going to look at what they're sharing together, which is SAD, you know, which is that read group counts. You know, these are the counts, read group counts here. You're just going to, that's what SAD is, you know, those counts. And you're going to just scale it instead by FA unique. You see that the numerator is always the same, but the denominator may change based on, you know, what you're sharing it with. So here it's FA unique and FD unique. So that's why the denominator is just going to be, you know, it's sharing it with D. So you take the sum of A and D, and then the numerator is just going to be FA unique. So that's how you get this fraction. So you notice the same numerator, but the denominator, so this part remains the same. It's just the denominator part is changing. What you're adding to it changes. And if you're sharing it with multiple groups, then you would just tack on the multiple groups to it as well. If you're sharing it with multiple groups, then for FD unique and then something else, you know, if they were like 3 pointing, like we're going to cover that example and I'll explain that there. But that's how you get these shared counts here. So then you'll take these values and you'll sum them up then. So yeah, that's how you get SAB counts and SAD, you know, you get this part, which is essentially what this part is telling you is it's giving you, what this part is giving you this, maybe I should use another color. What this part is giving you is it's giving you this part. It's basically trying to tell you the part of B that A is getting. That's what this thing is telling you here. And then the other part is this sort of like telling you the part of, you know, what's being shared between them, SAD, that A is getting, that you would give, you know. So it's just like kind of like you're imputing, you're trying to figure it out that, hey, you know, I noticed that, and these values, FA unique and FB unique and FD unique and all these other values we just calculated, they're sort of scaled. The unique values that each one got, the unique read groups divided by, you know, in some way by the transcripts, the transcript length. So it's sort of scaled, you know, we scale these values. Like 40 over 120 is 0.33. We scale the read groups by the transcripts. So we've taken that information into account when we're looking at FA unique and FB unique and FD unique. We're taking the read groups and the transcript information into account. And that's the best information we have to work on. Like, how do we allocate these read groups to these different transcripts? Like, it's very difficult for us to figure out, okay, well, out of 390, okay, so I know all these 620 belong to transcript 1, for instance, but how would I divide up the 390 between these? You know, how would I do that? Well, I'm going to use the best information that I have. I know that I have 620 unique read groups that go to transcript 1, which has this transcript length. So I'm going to calculate this unique, you know, F1 unique, which will be 620 out of divided by 40, and I'm going to get that value. And then I'm also going to, you know, see, okay, well, it has 390 that it shares with transcript 3, but how is transcript 3 placed unique in a unique way? So 5500 divided by 1000, you know, 5.5. So we don't, the shared part is almost like something we don't know. So it's like if you think of like a system of linear equations, you've got x and y and z, and you've got maybe multiple different variables. You try to solve for what you know for now, and then reduce the scale of the unknowns that you have to solve for. So we definitely want to know that these definitely, like the unique groups definitely map to one transcript. So we should use that to get some information, you know, some approximate idea for how to split up these values. So it's just like being a detective, using some past information to have some more knowledge on how to proceed and how to act and what to think. You know, how would we divide up these shared counts? That's always a difficulty. We don't know. Maybe we just split half and half, right, between them, or maybe not. But like, we want to be really accurate because, as I showed you guys, RNA sequencing data has so much, you know, power in helping us with scientific discoveries. So we should spend the time to do a really good job. So pretty much we know this UA here, that for sure belongs to transcript A. We're just trying to determine what part does A get from SAB, you know, when it shares with B, and what part does it get of SAD, you know, what it shares with transcript D. So we want to look at information about how B is, you know, using its unique read groups, and how D is using its unique read groups, and sort of scale, you know, SAB by you know, these relative unique abundance values. So, you know, this is the whole formula here for, you know, like, so this would be for the transcript A. So this would be for A. So for A, the C rescue, you know, this transcript for now would be UA and these values. And again, it's like an expectation maximization. So we're having some initial value here, and we're going to make some updates as well. So it's like more like an iterative process where we're always working towards getting the better result that we want. So now we just start updating the counts for each transcript that we have using this formula here. You know, we take the UA, that part is always belonging to transcript A, and then we're just scaling, you know, what it gets from transcript, you know, like from this read group SAB, what it shares with transcript B will be the scaled version. And then what it gets with, like, for SAD, which it shares with transcript D, would be, you know, this part here, this scaled part. You're just basically like divvying it up. So then if you think about if you had to ask, okay, well, what will B get from SAB? So if we were looking from B's perspective, B is going to then get the remainder, you know, which would sum up to 1 SAB, you know, counts. So it's going to get the SAB counts, and then you just have to multiply it by FB. I'll just write this for, like, you know, I'll make this FB divided by FA plus FB. These are like these approximate values, FB, FA. So I'm not writing the unique part though, FB. So that's what like B would be getting instead. And what you'll notice is that, you know, if you add this and this part up together, they will sum up to 1. So it's just like the same piece of the pie. It's just, you know, it's just you're dividing this SAB up into what A will get and what B will get. So maybe, you know, you just take this part here and we're just going to divvy it up. You know, what part will they get? Oh gosh. That's all you're doing here. And maybe A gets a huge chunk of this. You know, maybe this is the part that A gets. Maybe a lot. And then B gets the rest of it. Like, it gets all that remaining yellow is left for B to take. But that's sort of how you will divide up these read groups. So similarly, now going back to our example, so remember I mentioned that these are the values we have computed, you know, these are the values that we have gotten. So we had gotten our unique and values here from earlier, you know. So this will be kind of what we are going to work with. And again, when I had mentioned that we take the 40 and then we divide that by 120 and then we get a value, then we sum it up, and then these are the percentage values that we've gotten. So there was 40 out of 120. We got, and then we took like, just to recap again, how did we get these values? So we took the 40 divided by 120, we got like a 0.33. Then, you know, we got 5,500 divided by 1,000, 5.5, you know, 405 divided by 270. We got like another value, which is, just check. Yeah, we got another value here. We took that value, then we got like 620 divided by 40. We got another set of values, then 180 divided by 100, which is 1.8. We summed up those values as well, you know, which is 24.63. Then we took each of these values, like 620 divided by 40. When we had calculated that value, then we divided that by 24.63 to then get like 62.923. That's how we had gotten those values. So again, we took regroups divided by transcripts for each of the unique ones. We summed those values up, got the total. Then we took the regroups again by transcripts. We had divided them, and then we just divided them by the total to compute these values. So please let me know if you are stuck on these. But sort of like that idea is that we take the those unique values. So if you're trying to find the C rescue for transcript 1 in this example, because I showed you the toy example, so we would look at the unique counts for transcript 1. So the unique counts for transcript 1. Okay, so let's see. Okay, so the unique counts would just be this, you know, 620 value here, which is what we have here, the 620. Then the next thing is we're going to look at the shared counts of transcript 1. Okay, so what is it sharing? Okay, so what we see here is, okay, it's sharing this 390, but who kind of can have claim to this value? Well, transcript 1 is sharing it with transcript 3. So that's what we're going to focus on. So this is like your S13, it's like 390. So transcript 1 shares a read group of 390 with transcript 3. So what transcript 1 will get out of this will be S13, you know, the 390 value, and then we take what transcript 1's relative unique abundance is, which is 0.62923, and then we divide it by, you know, the relative abundances of transcripts 1 and 3, so whatever is being involved. So that's what we care about. So here, you know, 1 and 3 here. So that's what we care about. Transcript 1 shares it with transcript 3. That's why we have this here, 1 and 3. And, you know, this 0.623 value here, and then this 0.223275 is for transcript 3. Then this scaling is 0.738095. So that sort of tells you if we were scaling it, what value, you know, relative, we see that like, you know, 73.805. So if we just had to focus on just this right here on transcripts 1 and transcripts 3, and then we sum them up together, then about 73.8095, you know, about 73.8% of the relative abundances would belong to transcript 1. So that's what we'll get here, and later on we'll see when we're looking at this for transcript 3, it'll be the flipped around version. So we should expect maybe, you know, 1 minus this value, which would be like about 0.26 something to come up. You know, 1 minus 0.738 would be like 0.262. So then we just multiply this out, you know, the scaling factor like this. This becomes this value here. And then this is 287.8572. And that's what we get here. So we take the 620 and then we take this 287, we get a new value. So this 287.8572 and the 620, we're going to take both these values and then we're going to plug in this value and this value to get C rescue transcript 1. And that's what we do here. We just add them together, and this is 907.8572. So those are the updated counts for transcript 1. So we do the same thing for transcript 2. So let's focus now on transcript 2. So we notice that the unique counts is 405. That's for transcript 2. And so we get this 405 from, U2, like a great band, huh? And then we're going to look at the shared counts of transcript 2. So I wanted to point out something here, that what we notice is that, okay, well, we have this 4100 value here, but where is it coming from? Well, it's being shared with transcript 2. It's also being shared with transcript 2 has it, but it's also sharing it with transcripts 3 and 4. So it's sharing it with multiple read groups, so not just one. So hopefully this will point out any questions you might have on a more complicated example, like what if you have multiple read groups that it shares it with? Yes, you have to just scale it relative to the other groups, right? So many transcripts are like competing for the same read group, right? So you're going to use the information. How do you figure out how to divide this 4100 among 3 read groups? And this is why it gets more complicated. So that's why you're going to figure out, okay, well, what is the unique transcript abundances? And you're going to use that information. So we see transcript 2, so S2, 3, 4 is why I've called it. It's shared among transcripts 2, 3, and 4. So this value, this 4100. So whatever transcript 2 will get will be its relative unique abundance in the numerator, and then its denominator will be this unique for 2. But also you're going to be adding it up for the groups it's sharing with, which is 3 and 4. So that's where we get the denominator. So the numerator part here, this F2 unique part here, will be here, and then it's also coming here again. And then the other parts that it's sharing with is just going to be this unique parts here. We're just going to take these values here. Okay, these values right here. And that's what we will put here. And you'll notice that these are the same, values here that we've gotten. And this would be the part here. And this part here is the 4100 that we have over here. And yeah, that's what you'll see, that we're just going to, we sum these up, we get like 0.2977, and we're just scaling it. And then that's equal to 4100 times 0.204545, and then we get 838.6339. So that, is going to help us. So then what you're going to do is, now that we found, 405 again is the unique group for, transcript 2. So, we have this value here for transcript 2. This is the the U2 that we got. And then the shared count, we just found right now is 838. So out of this, 4,100 that's shared between the 3 groups, so we get about 838 of them go to transcript 2, which if we sort of compare the values for groups 2, 3, and 4, could be a little bit surprising. Why does it get such a low number out of 4,100? Well, that can be seen maybe by these relative unique abundances, where, where like transcript 3 almost has over 3 times as many as transcript 2, and we would expect a lot less for transcript 4. So let's sort of see if that matches. But keep in mind that out of this 4,100, we're just getting like 838 coming to transcript 2, out of this value here. So that's sort of like what it's getting from here. And a lot more should be going to transcript 3, which we'll just find out right now. So we just take these values and we just find that the updated counts for transcript 2 will be 1,243.634. So that is the updated counts for transcript 2. So next we're going to, so I'm just going to make this, since we're doing the same process again, but now we're going to focus on transcript 3. So what we notice is that transcript 3 is involved in these 3 different read groups: 390, 4,100, and 5,500. So we notice that, okay, well, which groups really belong to, so same thing again, we notice for transcript 3, what it really has to itself is this 5,500. So that's quite a bit of, of reads, right? 5,500. Okay, great. So it's got a lot of the reads, but it shares 390. So it shares this. So this is the groups that it's sharing. So it's sharing, 390 with, transcript 1. It's sharing this. They are sharing the 390. And then we're also finding that we're calculating for transcript 2, we find that it's also sharing 4,100 with these other transcripts, 2 and 4 this time around. So that's what we're finding. Okay, well, it's got 5,500 to itself, so that's this count here. And now we just have to figure out, okay, how do we divvy up? How do we divide 390 and 4,100 among it? So we're going to do the same thing for 390. Instead, now, before we were looking at F1 unique over F1 plus F3 unique, but now because this is all about 3, it's all about this 3 over here. You see, like, everything is about 3. So maybe let me highlight that here. Yeah, so this is about this 3 here. 3 always has to come here. It's all about 3. So that's why we take the 390, and then we kind of take, F3 unique and divide by F1 unique and F3 unique. That's what we're scaling it by. And that's where we get this fraction. But we should notice that, before when we were looking at, C1 rescue, it was, 0.62923 in the numerator, and the denominator was the same, but the numerator was different. But here we're figuring out what part do we give to 3. So that's why we have to scale it this way. And then, so again, ask me if you have any questions, but, we looked at this for the first part. So please check that out again, for more clarification. And always ask me if you have any questions at all, please. So there's 4,100 here. And then we're just taking this F3 unique, which we have as, 0.223275. And that's what we're putting here. Again, you can see that is just the same, right? Like, these values are, this is the same as this, right? It's just F3 unique. You're just taking that same value, and you have this here, you have it here, you have it here. It's the same value here, and you're just scaling it by the other part. So this part here is helping you out with this part, from the diagram that we saw. This top part is, all of the red edges, and this other part is all these blue edges here, right? So this is divided by, what group transcript 2 is getting, what part of the, overall same group that it's getting for 4,100. You get it from this 0.60893. Then this part again is, for the relative unique abundance for transcript 3. Then this 0.013532 is coming from transcript 4. So that's kind of, in the end, as we predicted, the transcript abundance, the unique abundance of transcript 3 is high. So that's why, like, when we simplify this value, it's going, the fraction, the same value from before, you might remember 0.2977. A lot of it is going actually to, unique transcript 3. Transcript 3 has a lot of it. 75% actually, that's what this maps to, this part here, what this maps to. Maybe I should use a different color. Well, yeah, but this part here is mapping to what this part maps to right here is just going to be this 0.75. So it's going to get out of the 4,100, instead of getting like 800 something, it's going to just get 3,075. Not just, but that's a huge amount out of these 4,100 reads. 3,075 of those reads will be given to transcript 3. That's what it's going to get out of this. And around 800-ish was given to transcript 2. So the remainder, as you could see, will be given to transcript 4, which has a relatively smaller unique relative abundance. So then again, so we just take this 5,500, these, counts that just map the read group that just maps to transcript 3. Then this part here, comes from this part, from this part, from this group this 390 group, it gets 102 out of it because transcript group 1 takes most of that out of the 390. It takes most of those reads. So it's just transcript 3 is just left with 102 out of the 390. And then here you take this part here, which is out of the 4,100. What part of it is given to transcript group 3? So you take all these values and sum them up, and then you get 8,677. So the updated rescue count for transcript 3 would be 8,677. So this is sort of, kind of showing multiple things here. Showing what happens when you, like the same thing as before, when you're having a read group that's shared between, with multiple transcripts, like 2 others, which we also showed for C2 rescue, but this one part alone shows both things together. So it's sharing with multiple transcripts, and it has multiple read groups that it's sharing. So it, this 4100, what this example is showing you is that it's, transcript 3 sharing it with 2 other transcripts, and you will do the same thing for additional ones. You just have to take whatever the transcript is in the numerator, divided by that transcript again, and by along with the denominator includes that transcript and any other transcripts it's sharing with. You're just going to add up the relative abundances. So if it was sharing it with X as well, for some reason, if there was an edge that was going from here to here, you would just have to add in this X part here, this Fx in the denominator, and so on. Like you just have to add it to the denominator. So then this would be your new denominator. Your new denominator would be all of this. This is your Fx in the denominator. Oh gosh, I messed it up. Okay, but anyways, your denominator would have this Fx part. So like if you just had an imaginary edge going there. So what I wanted you to just see is that from that example is just, just kind of seeing again this idea of, sharing between multiple groups with the 4100, and the idea that it also has like many read groups that it has, not just the one it's sharing, not just one additional one, but multiple read groups. You just have to add everything together. That's what I wanted you to take from this. And then like for transcript 4, you do the same thing. It's a little bit simpler now when you think about it, but we have this 4100, and you see this value over here that is being shared. It's sharing this 4100 value here, and this 40 is being unique. And then we can just do the same thing again. We have these 2 read groups, 4100 and 40, and read group 4100 is shared with transcripts 2, 3, and 4, and then these would be the updated counts for transcript 4. And last but not least, the last guy, which is the CX rescue for this last transcript X. So what we're going to see is that it's just the only person that maps to it is itself. So it shares it all to itself and got nothing to share with anybody. So in the situations where nothing is being shared, your CX rescue is just 180. So the same counts, just stay to itself. So you can sort of think of it in a way as sort of like splitting up what is it getting from each part. So before we take in, like when we see this, what we see is is the following. In general, guys, what you'll see is you're going to have like a transcript here, like, that it's sharing. And then you'll find that, hey, okay, well, it's got this 620 read group all to itself. But then what we're going to essentially add, what we're adding over here is we're adding on the information about what part does it get out of this 390. So if we had to look here, we'd say, okay, well, this is 390, it's sharing this 390. What part does it get? So when we figured out, we used the relative abundances. We looked here at F1 unique is 63% relative to what, the third group is having, which is this value of, 22%, of the relative abundance. So what we'll say is, okay, based on that fraction, how do I split up this 390? Well, maybe I'm going to take these two values and then figure out like which percent, like, take the percent of this percent, add them up together. And then we have 0.629 divided by the total of F1 and F3 and get that value. So this would just tell us the following. What this will tell us is the fact that out of the 390, we're just going to end up getting like 73.38% of that. So when we take this value, relative value of this, we end up getting that for F1 unique, that weightage just becomes like 73.8%. And then we multiply that by the 390, and then the total weight that we end up getting is 287.86. So that's the total value that we end up getting here. So then when we add them up together, what we will end up getting is 907.8572. So we end up getting this value here, which would be the total count for transcript 1. So again, this comes down to us having for transcript 1, the C1 rescue. We'll say that the updated counts that we have for transcript 1 will be 907.8572. So that's how we get those updated counts. So that's, I hope that this will help you guys see so far. So like if there's nothing being shared, then we just assign that to CX Rescue. So sometimes I feel like you might think that, okay, there's all this stuff to memorize, but no, it's just the concept. The concept is we just really want to know what part of these read groups belong to it. We want to figure out, okay, well, what part do we actually give to this group out of this? If we have this part, what part do we give? We know that we can give this whole part here to, transcript 1, the whole thing, but what part can we also give to it from the shared? That's all we're trying to find. And then when we take that part, the contribution, this will be the part that belongs to transcript 1. We just really want to essentially break it up into the graphs where we have its own part, the part that belongs to it. So if we could, we would just get rid of this shared part and just have the 620, 287.86. Then we would have what transcript 2 has, and we wouldn't be having any of these shared edges. We would just get rid of it and just say, instead of having the shared edge between these two, I'm just saying that this is 620 belongs to transcript 1, this 287 part belongs to it, and then I would have for transcript 3 the part that it has instead. So that's what we're trying to do. Just figure out what belongs to it by itself, have those updated counts. Not really the shared parts are just a little bit worrisome for us. So we're just trying to figure out how do we divvy them up. And the best way for us to divvy them up is to go off of information that we know, which is the unique ones. The unique ones we definitely know belong to a transcript, to that particular respective transcript. So we use that information to sort of guide us. We use those fractions and relative values to each other to guide us for how to split up these shared counts, these read groups that are shared between multiple transcripts, 2 or more. So these are the values that we'd gotten. So the 180 was kept from before. We had the transcript length for each of these transcripts. These are the rescue values that we calculated. The rescue sounds like very heroic, right? And that's what you get these values here, so just a little bit more simplified. Then you can sum up these values here. When you sum up all these values, you're going to get, just sum up all these values and you'll get like the total, which is 39.666. And you might be noticing a trend that maybe what you might be noticing is a trend where, this 1.88 as well as being there. You might be noticing a trend where we take these values and we're just summing them up together to get 39.666. So all these values summed up will give us this value here. So again, this is just a simpler version again of just seeing that we take these values and we just sum them up together, get this value here. And we're just going to do the same thing, which you can probably predict, scaling it again. So we have these values, we have a total number, so we want to update the relative abundances for each transcript. So you can think of it like it's a very iterative way, just we had these initial values, if you remember, that we had just used to update our counts. We have some count data, we looked at the unique counts, then we'd gotten these updated abundance counts for the unique reads. And then we use that information to update our counts as well, like split the ones that were shared by multiple transcripts. And now we're just going to, again, update these values, these relative rescue values. We're just going to update them. And this time we're going to be considering not just the unique ones, which is what we did the first time. These C1 rescue values we looked at before the first time, those were just unique. We had looked at just the unique ones, and then we had gotten these F unique values. And then we use those to help figure out how do we split up the read groups that are shared by 2 or more transcripts. Now we have figured out, we've updated the counts. So then what we're going to do is use this updated counts information to update these F values again. So it's like EM, it's like you get some value, use that to get something else, then use that to update the previous one, and it's like very back and forth and back and forth and back and forth and back and forth and back and forth till convergence, So we're just using like one iteration of it, of expectation maximization. So we have, this is like these values that we had computed, we have the total. So what we're going to do is we're going to say, okay, well, we're going to just basically take this value that we just found, and that's what this is, and we just divided by the total, which we had found is 39.666. We just divided by the total, we're making it into a percent like we did before. The same idea, we have this value, we divided by the total here, and we have a percentage, which is 57.21. I think before it was like 62.9%, and now it's now it's scaled down to 57.21. So that's what F1 rescue is. So we're going to do the same thing for the others. We're just going to take, we have this total value here, that's 39.666, and then we have this, all these values here, even for the last one as well. We have this value here, then we have like this total that we're using, this total, and then we're just using here this 22 value that we just saw. Here is like 4.6. We just take this, we divide it, we get our value. Is 8.66. This is where we're just taking these values here, this value here, and that's how we, and the same thing for this guy over here too, and that's how we're getting the numerator as well. So that's how we get these values. These are the updated values. So now they're scaled again. But these are the updated rescue values we got. And this is how we can update the relative abundances for each transcript. So all in all, when we're asked to use the RNA rescue method to calculate the relative abundances for these 5 transcripts here, this is the values that we have gotten using expectation maximization, one iteration. So that's sort of how we can go about and calculate and solve this problem, and I hope that this has made some more sense to you. Please let me know if you have any questions at all, and I think maybe we can try to make it like a Superman in a way. I'm kind of bad at this, but okay, like let's say there's like a little logo in the back, it's like RNA sequence discovery, and we could try making it like a little Superman. I used to like to sketch this a lot. Okay, great. And it's like, so this is how you can go from, this transcripts and read groups, figure out how they're shared, and then come up with the relative abundances that would be very helpful. So this is this first part, and just to summarize it up, you can see all these numbers, all these characters. This is how we can go about it. So first step is please represent graph as a read groups, read groups to transcripts. Please then find the reads and transcript lengths for uniquely mapped reads. Please connect unique mapped reads, transcripts, and transcript lengths together. Then find the unique read group for a transcript divided by the transcript length for each transcript. Then you find the unique transcript relative abundance values. Then you update the counts, for each transcript. Then you divide the updated counts by the transcript length for each transcript, and then you can update the relative abundances for each transcript. So it's a very iterative process that's known as expectation maximization. So again, guys, my name is Saniya. If you found this was helpful, please like my video, and please let me know if you have any questions at all on this first part of the RNA sequence rescue method. Thank you, guys.